Welcome back to Dr. Liu's VCE Biology. Today's topic is photosynthesis, cellular respiration, and biotechnological applications, as presented in the 2024 Victorian Certificate of Education, VCE Biology exam. Let's learn together. If teachers, parents, or students notice mistakes in my explanations, please provide me with your feedback. The Krebs cycle, also known as the citric acid cycle, is a crucial part of cellular respiration. It occurs in the mitochondrial matrix, location 2, and is responsible for producing a low amount of ATP, 2-ATP, during aerobic respiration. Cellular ATP synthesis occurs at different rates in various locations. The inner mitochondrial membrane, or cristae, location 1, is where the electron transport chain generates a substantial 26 to 28 ATP. Conversely, the mitochondrial matrix, location 2, home to the Krebs cycle, and the cytosol, location 3, where glycolysis takes place, each produce a modest 2 ATP. The outer mitochondrial membrane, location 4, yields no ATP. A common challenge for students, particularly those who struggled with question 8, was distinguishing these sites. They may have mistakenly identified the outer mitochondrial membrane, location 4, as the location of the electron transport chain, overlooking the fact that this high ATP producing stage occurs at location 1. A biochemical analysis of bat pancreas cells showed a high level of lactic acid. Since lactic acid is a product of anaerobic respiration, also known as anaerobic fermentation, in animal cells, and anaerobic respiration occurs when oxygen is absent or in low concentrations. This result directly suggests that oxygen levels are very low or absent, leading to anaerobic respiration in the bat pancreas cells. Occurring in the inner mitochondrial membrane, cristae, the electron transport chain, etc., is a critical stage of cellular respiration. It produces a significant 26 to 28 ATP, and its outputs also include water and ATP. NADH contributes by donating electrons and hydrogen ions. This stage is fundamental for generating the energy required for various cellular processes. This experiment examined the influence of various light conditions on the cell density of photosynthetic microalgae. Under white light conditions, cell densities demonstrated a consistent increase across all measurement days. Notably, blue light conditions produced the peak cell densities by day 6 when compared to the other light spectra. Conversely, green light conditions were associated with a reduction in cell densities from day 3 to day 9. The light intensity of all the different lights, option C, served as the correct controlled variable. Conversely, option D is incorrect. Natural cellular variation means the microalgae in this experiment would exhibit slight differences in the relative amount of chlorophyll molecules per algal cell. The light energy absorbed by microalgae cells is immediately utilized in the light-dependent stage of photosynthesis, which occurs at the thylakoid membrane, grana, making option A correct. In this crucial process, water molecules are split into oxygen, hydrogen ions, and electrons, the latter two collected by NADP+ to form NADPH. Conversely, option B is incorrect because the light-independent stage occurs in the stroma, and does not directly depend on light energy. Students must understand the locations, inputs, and outputs of the various stages of both photosynthesis and cellular respiration. For photosynthesis, carbon dioxide enters chloroplasts via dissolution in water. Nevertheless, an elevation in temperature diminishes carbon dioxide's solubility, thereby reducing its propensity to bind with rubisco. Consequently, the rate of carbon fixation is slower at 40 degrees Celsius than at 20 degrees Celsius, validating option C. Option D is incorrect, as increasing temperatures are associated with an increased likelihood of oxygen binding to rubisco, not a decrease. Bioethanol is produced by yeast and microorganisms through fermentation, a process that also yields carbon dioxide. Cellulose, a key component of plant cell walls, serves as a suitable biomass source. Option C is incorrect because biochemical pathways utilize distinct enzymes to catalyze each step. As fermentation progresses, ethanol concentration rises and glucose concentration falls. Yet, glucose often doesn't fully deplete even after 500 hours. This is likely due to several limiting factors, including the cessation of anaerobic respiration, the requirement for more time to consume all glucose, the toxic effect of accumulating ethanol on yeast cells, or its impact on pH, yeast cells potentially having met their ATP demands, or other constraints such as the number of yeast cells or the amount of coenzymes available. Adding oxygen could lead to less or no ethanol being produced. This would occur because aerobic cellular respiration would take precedence, leaving less glucose available for anaerobic fermentation, which is the sole pathway for ethanol production. Photosynthesis is considered a biochemical pathway because it involves a series of reactions, light-dependent and light-independent stages, where the outputs of one reaction serve as inputs for the next. It relies on enzymes, like rubisco, and coenzymes such as ADP, ATP, and NADP, NADPH, with different reactions occurring in distinct locations, grana-thylakoid membrane and stroma. Furthermore, its regulation is influenced by the availability of reactants, concentration of products, and abiotic factors. This slide poses a question about C3 and C4 plant photosynthesis, specifically their carbon dioxide fixation under varying light conditions. The graph clearly shows C4 plants consistently fix more CO2 than C3 plants at all light intensities. In response to the sub-question, this data demonstrates the competitive edge of C4 plants in fluctuating light conditions. Emphasize C4 photosynthesis efficiency, enabling them to thrive alongside C3 plants, especially in high light. Justify your argument with specific data points from the graph. This slide aims to highlight the competitive edge of C4 plants over C3 plants, with a strong emphasis on data-driven argumentation. Encourage students to use specific figures, such as the carbon fixation rates. For example, C3 plants fix 90 mmol/hour at 400 lux, while C4 plants reach 150 mmol/hour. This demonstrates that C4 plants achieve greater and more rapid CO2 fixation, which translates to superior photosynthesis and growth. The provided mark distribution table illustrates how data accuracy directly impacts student performance, while common misconceptions, like confusing CO2 uptake with fixation, point to areas where clarity and precision are paramount. This slide asks students to explain the relationship between light intensity and co-fixation in C3 plants, specifically why fixation rates change and then level off beyond 1700 lux. The accompanying table details key influencing factors, such as the efficiency of the light-dependent reactions, and Rubisco enzyme saturation in the Calvin cycle. While many students noted the plateau, they often missed explaining the initial increase in co-fixation with rising light. This initial boost occurs due to enhanced energy production and more substrate for the Calvin cycle. The marking scheme emphasizes a comprehensive answer, highlighting that both the initial increase and subsequent leveling are crucial for understanding photosynthesis dynamics. The slide delves into C4 plants' superior ability to minimize photorespiration compared to C3 plants, particularly in warmer conditions. This efficiency stems from a unique mechanism that ensures Rubisco has a greater affinity for CO2, thereby reducing its binding with O2. Furthermore, locating CO2 fixation within bundle sheath cells effectively limits O2 competition during the Calvin cycle. Key misconceptions to address include the mistaken idea that bundle sheath and mesophyll cells are spatially identical, or that C4 plants exhibit CAM-like diurnal stomatal closure. The provided mark table highlights the necessity for students to perform a clear comparative analysis of C3 and C4 plants to earn full marks. The 2014 VCE exams focus areas in photosynthesis, cellular respiration, and biotechnology are summarized on this slide. 5 key topics are highlighted: the Krebs cycle and ATP production in cellular respiration, the significance of light conditions for microalgae in photosynthesis, the role of enzymes in biochemical pathways, and the advantages of C3 versus C4 plants in CO2 fixation. Additionally, the slide identifies the question format, multiple choice, or short answer, for each topic. This structured overview is crucial for students to grasp the exam's demands and prepare effectively for potential questions. This slide breaks down question types and their marks, essential for understanding the assessment structure. A score of 20% was achieved, representing 24 out of 120 total marks. This percentage indicates performance and guides future improvements. The table categorizes questions as multiple choice or short answer, each with specific marks. Short answer questions are further divided, suggesting a more complex evaluation. This detailed distribution helps analyze which question types require more focus in teaching or revision. If you need my help, please add me on WeChat or WhatsApp and send me an email. Thanks for your attention. Please note Dr. Liu's VCE Physics channel, Dr. Liu's VCE Chemistry channel, and Dr. Liu's VCE Biology channel.